So in the last part, we have studied about vector. So vector is the carrier or it's the vehicle to carry our gene of interest to the host. So today, now we are going to study what are the ideal features of vector. Features of ideal vector. In the last session, just I concluded what are the points I have given. But today we are going to study detail what are the features of ideal vector. So first one you can see here, first feature is ORI or it is called origin of replication. As we know, it's a sequence of DNA. Origin of replication is a sequence of DNA where the replication starts. So here the origin of replication where the replication starts. Replication starts, or you can say it is used to control the copy number. How many number it has to make cloning, replication. So it is used to control the number of copies. So that only you can say origin of replication. So it is used to replicate, control the number of copies. It is called origin of replication. So in the ideal vector, this is one of the parts or it is one of the feature. So second one you can see here, the second feature is selectable marker. Second feature is selectable marker. So what you can understand about the selectable marker? First of all, we can understand the term, one of the term which are using in the biotechnology, that is transformation. The term it is called transformation. What is transformation? Transformation is the process where the gene of interest or the DNA it is inserted into the host. The DNA it is inserted into the host, that is called transformation. After the formation, after the transformation, there are three possibilities can take place. That is, first one you can say, first one is recombinance, or it is called transformance, and the second one is called non-recombinance, or it is called non-transformance, and the third one is transformance, but without RDNA, without RDNA. So what you can understand here, after the process of transformation, there are three things can take place. That is recombinance or transformance. The another one is non-recombinance or non-transformance. The third one is transformance without RDNA. Here we need only the recombinance or transformance. So we have to identify, we have to understand only the recombinants and eliminating non-recombinants. So by the help of some selectable marker, by the help of antibiotic resistant gene, that only we can say selectable marker. So selectable marker is used to select recombinants and eliminating non-recombinants and allowing the growth of only the recombinants. That we can understand about the selectable marker. So selectable marker is used as an antibiotic resistant gene. So some of the example for antibiotic resistant gene, tetramycin, ampicillin, keramycin, chloramphenicol, these are the example for selectable markers. Selectable markers are used as antibiotic resistant gene. So that you can understand, means it is used to select or it is used to identify only the recombinants. We need the recombinants. So we have to identify the recombinants and eliminating non-recombinants and permitting the growth of only the recombinants by the help of selectable marker. Next, third one we can understand recognition site. Third feature is recognition site. So what you can understand here, the recognition site, as we know, one of the plasmid we are using in the biotechnology, that is PBR322. PBr322. So this is one of the plasmid we are ever using in biotechnology, that is PBr322. So in the one side you can understand ORI, and there are two antibiotic resistant gene is there, that we can say here it is tetracycline, and here it is ampicillin is there, and in between here it is BAM H1 and eco R1, and here it is PEP. So there are recognition sites are present. So these recognition sites are used to cloning our gene of interest, So in the recognition site should be there one or two, means it should be few in a ideal plasmid. It should be very few in the ideal plasmid. Otherwise, there are many segments have to be cloned, and it will be complex. It will be the difficult process. So it should be one or two. It should be few. So that you can understand about the recognition site. So in the next video, we can understand what the detail about the recognition site. Thank you, students.